So next comes the questions, how long should be the ORF? So if we look into theory, there are 64 possible codons, and there are three, the length of each codon is three nucleotides. So these are actually triplets. So in this way, we can have 21 codons. We can expect 21 codons before we see a stop codon. So in that case, our ORF length may be this much long. But is it the right length? Obviously no, because mostly we know that our genes are much longer than this. So we might scan longer ORFs than some threshold sometimes. So an ORF is more reliable sometimes if we look into the frequency of some expected codons, we can put more reliability. So ORF is more reliable if it has likely codons. So what we can do here is we can make a sliding window. So we can focus on some nucleotides, like for example, if the segment is 1000 long, so we can make a window of say size like 50, and we can look into the frequency of different codons in that window. So we can look into what in these windows, what is the frequency of those likely codons which are expected. We can get an estimate from while looking into the protein product of that particular gene also. So, however, there is one more thing that in case of the eukaryotes or in case of the vertebrates, the exon length is a little bit small. So as far as the introns, they are much longer. So that creates an issue. So those kind of generating those sliding windows and do these codon usage tables, or finding those likely codons becomes complicated. So we might then break up the fragments into hexamers, two codons at a time. So we can look into these presence of these six units or six nucleotides together, or two codons together, and we can have a comparative ratio of different hexamers or frequencies. So that can support in order to correlate those ORFs to the true genes. Tools which are defined to identify ORF, they are called as ORF finders. So there are three famous tools. One of them is NCBI's ORF finder. Then there is ORF investigator, and there is ORF predictor. So I will go and elaborate a little bit about this NCBI tool because it is more frequently used. So if you go to NCBI website, it is there. It's called as ORF finder, Open Reading Frame Finder. So you can put your sequence into this space over there. So here you can upload your sequence, and then you can just click this ORF find. You can clear your sequence. Sometimes you can try to enter the gene number or accession numbers here, and it will get the sequence by itself. So when you click ORF find, it will give you these different reading registers. So there are six of them. Top one is from the top strand, and then bottom one is coming from these. Lower three are actually the negative registers, and upper three are positive registers. So if we focus on any one of them, it will then highlight you about the actual sequence which it got. So it says that it got on position number 1, there is a start codon ATG, and that is also indicated here with this small dot, or like half this line like thing. And these pink lines, they tell you about those stop codons. In this way, we get this ORF whose length was 54 nucleotides. Now, same way, it can also give us the protein it is encoding. So as you see that ATG is encoding methionine. So that is the case which is true for almost all of the proteins. So the first codon is ATG, and where we have the stop codon, we will observe that steric. So here it means that our translation, our protein synthesis stops here. So you can same way click on rest of those frames or registers, and it will highlight what sequence is there in this bottom section. So ORF provide important evidence as far as the gene finding is concerned. Generally longer ORFs are preferred, but ORF most of the time they may not correlate into the proteins, and ORF finders are the softwares which are based on mainly pattern finding algorithms.